Hi everyone, my name is Jack Kincaid. I'm a fourth year student at Case within the Department of Nutrition, and today I'll be talking about flow cytometry. Here I have a number of cells, these are in green. Flow cytometry is a technique that is predicated on the idea of differential expression of not only intracellular molecules, but also cell surface molecules like the antigens shown in blue and yellow here. So flow uses concepts from fluorescence spectroscopy. So we take antibody molecules that are targeted to these specific antigens, they'll attach, and these antibodies are labeled with a fluorescent tag, either a fluorophore or a fluorescent dye. And once those are excited by a light source, such as a laser, they emit fluorescence that can then be used to discriminate between each of these cells. But why is it called flow? So cytometry is the analysis of cells. Flow cytometry is sort of talking about this flow of cells. So when they're introduced to the machine, they're in a suspension. So here we have a group of cells. Sorry for my bad drawing, but a sheath fluid, this is a microfluidic concept where a sheath fluid is accelerated around the cells at a rate of about 5 to 10 meters per second. And as it's accelerated, it pushes the cells in and pinches them into a single file line so that they can be interrogated one by a laser. This maximizes resolution and minimizes noise of other emissions from other cells. Let's look a little bit more at the apparatus. We show the cells here in the suspension and being pinched by the sheath fluid, and now it's being interrogated by this laser, which emits light at a specific wavelength. Some machines have multiple lasers, in fact, to target different colored dyes. And once the light is shown onto the cell, fluorescence is emitted as well as light is scattered. So there's side scattered and forward scattered light, and that light is taken up by photomultiplier tubes. So before that, it's separated based on wavelength by a series of dichroic mirrors in this photo-optical emission system. And then the corresponding light is received by these photodetectors. The voltage from these photodetectors is digitized and sent to an electronic system for computational analysis and something called gating, which I'll talk about later. And after this analysis, the cells can sometimes, with certain machines, be sorted using this piezoelectric system, which charges cells and deflects them into a corresponding collection tube for further assessment of specific subpopulations of cells that are identified in this computational analysis. So let's look at what data is commonly shown with flow cytometry. On the left, we have a univariate histogram. So on the y-axis, we have number of events, and on the x-axis, we have the intensity of an antibody, in this case, A. So here we see two distinct populations of cells. A negative cells, which have low intensity of emissions being emitted from antibody A, as well as A positive cells. So these cells express antibody A. They are therefore bound to antibody A, and the fluorescent dye attached to antibody A emits light, causing this intensity to go up. And we can see that this group of cells, this blue slope, has the A expression. More commonly in flow cytometry, we use dot plots. Each one of these dots represents a cell, and so these are sort of groups of cells, and they're separated via a concept called gating, as aforementioned. And so this plot is quadratically divided to show different populations of cells based on their expression. So on the y-axis, we have the intensity of antibody A like before, and then on the x-axis, we have a new antibody, antibody B. In the bottom left, where we have cells with low intensity of both antibodies, there's no expression of A or B, so they're A and B negative, whereas in the top right, in this bivariate dot plot, we have high expression of both, so they're A plus, B plus, antibody A positive and B positive. This can be applied, this technique, in a variety of situations, but here I show it within a cancer context. This is the myeloid cell lineage, which is a big part of our innate immune system, and it's key to fighting off infection. And any one of these cell types can become malignant. In the case of acute promyelocytic leukemia, or APL, the promyelocyte malignantly transforms and becomes cancerous, and that's really bad. And so we want to study it in vivo in various animal models, and in order to do that, we have to be able to differentiate between each of these cells because we really want to focus on the promyelocyte. So here's an example where someone has created a flow analyte panel with these 5 antibodies that are differentially expressed, plus meaning expressed, minus meaning not expressed, in a variety of these myeloid cells. And then they can discriminate and find the promyelocytes. So in this case, it's population 2. And in this dot plot, we see population 2. So they're able to separate and figure out which of these cells are promyelocytes. Thank you for watching. This technique is obviously very versatile and highly applicable, and it's a very robust biophysical tool that will be in use for years to come.